So let's start this lecture, the excitation-contraction coupling lecture. So this is an overview of the objectives that we're going to do. We're going to start with the muscles and overview of muscle and muscle cell components. Then we're going to move to thick and thin filaments, myosin and actin filaments. Then we're going to go to cross-bridge cycling, then excitation-contraction coupling of skeletal muscles. Finally, we're going to go to the steps of the excitation-contraction coupling in cardiac muscle, because we're going to cover skeletal muscle first. They're very similar. And then we're going to compare skeletal muscle and cardiac muscle, because I felt like that was a really good point of really understanding what the excitation-contraction coupling is like. And I also noticed that your professor had a couple of slides on it in his lecture, so I wanted to add that to my lecture as well. Once again, this lecture covers the most important things that you need to know from your lecture. I did add a few things as well, because I felt like, just for the sake of completion, I wanted to add everything, so I just did that. But aside from that, it's pretty straightforward and it should be simple, inshallah. Okay, so first thing is the overview of a muscle and muscle cell components. Okay, so what is muscle? Muscle is a soft tissue that's primarily composed of long muscle fibers made up of muscle cells. These muscle cells are also called myocytes. And a single myocyte is the functional unit of a muscle fiber. The interaction of myofilaments, actin, and myosin within these myocytes is what gives the tissue the ability to contract. We're going to come to what actin and myosin are in just a minute, but essentially you can think of them like this. They overlap, and when they come together like this is when a contraction happens. When they release is when the muscle expands or relaxes. Contraction, relaxation, contraction, relaxation. Okay. So depending on how the myofilaments are arranged, muscles can either be striated or they can be non-striated. Striated muscles are cardiac muscles or skeletal muscles. Non-striated muscles are smooth muscles. Okay. So the filaments of striated muscle are arranged into sarcomeres. What is a sarcomere? It is the repeating unit of muscle tissue. And the myofilaments of smooth muscle lack specific arrangement, so no sarcomeres. So what is the purpose of muscle tissue? To produce a uniform or a collaborative muscle contraction together to create muscle tension and essentially stimulate movement or cause movement of the bones, skeletal muscle, the bones that are attached to the skeletal muscle. It's a skeleton, I guess. Okay. Coming to the components of a muscle cell or sarcomere, as you will. We're going to cover each of these in a little bit of detail. So starting with the sarcolemma. The sarcolemma is really just the muscle cell membrane. And this is what it looks like. This is a sarcolemma here. Think of the sarcomere as a cylinder, okay? We're going to keep that an image in our heads. It kind of makes it easier to picture. So this is the outside of the cylinder, sarcolemma. Sarcolemma has tiny holes on it. These are called transverse tubules or T-tubules. Each transverse tubule goes down into the cell where it connects, or it ends up being very close to something called the terminal cisternae. So these are the terminal cisternae here. And as you can tell, they're very close to the T-tubules. The T-tubule is an extension of the muscle cell membrane. Cell membrane is going down inside towards the terminal cisternae. And the terminal cisternae are extensions of the sarcoplasmic reticulum. So essentially, the T-tubules are extending from the cell membrane, and the terminal cisternae are coming from the sarcoplasmic reticulum, extension of the sarcoplasmic reticulum, and they both come very close together. This means that the cell membrane extension is able to touch an extension of the sarcoplasmic reticulum. They have communication there, okay? So this sarcolemma receives nerve impulses from nerve endings. So this could be neuronal nerve endings, or in case of cardiac muscle, which looks exactly the same, by the way. In case of cardiac muscle, this is a impulse from the action potential, or the pacemaker cells, sorry. Okay. So, these are the T-tubules, and sarcoplasm is the cytoplasm, the muscle cell. Okay, so T tubules that I highlighted over here, they invaginate into the muscle cell at the level of the Z lines. We'll see what the Z lines are in just a minute. And they're located close to the terminal cisternae, part of the sarcoplasmic reticulum, and they encircle the terminal cisternae, encircle the entire myofibril. The function of these T tubules is to transmit action potentials directly into the sarcoplasmic reticulum. Okay, so any action, the action potential that arrives will touch the sarcolemma and then be transmitted through the T tubules to the sarcoplasmic reticulum. I hope everything is clear so far. Okay. Coming to the sarcoplasmic reticulum, it's the same thing as endoplasmic reticulum. It's just the endoplasmic reticulum of a muscle cell. The sarcoplasmic reticulum has potassium release channels called ryanodine receptors on its surface. And the T tubule has dihydropyridine receptors on its surface. Here's an image that shows that. So this is the T tubule, which is connected to the sarcolemma, which was what? The external cell, the cell membrane surface. That's the sarcolemma, and this is a T tubule, and it has a receptor called a dihydropyridine receptor, or a DHPR, okay? This is the sarcoplasmic reticulum, which you'll notice has a receptor called the ryanodine 1 receptor, and these two are located very close to one another. Okay. All right. So what is the most important function of the sarcoplasmic reticulum, or one might say the only important function? Not the only. Okay, let's say the most important function. It is to store calcium ions that are later used in muscle contraction. Okay, so we're going to talk about how these two receptors that we see are connected, what they do, and how this eventually opens to allow calcium to come out of the sarcoplasmic reticulum. Okay. All right, so T tubules are connected to the sarcoplasmic reticulum via the terminal cisternae. They're able to very quickly transmit an action potential from the surface of the myocyte to the sarcoplasmic reticulum. This allows the sarcoplasmic reticulum to quickly release any stored calcium for muscle contraction. As soon as the impulse arrives on the sarcolemma, goes to the T tubules, goes to the terminal cisternae, goes to the sarcoplasmic reticulum, boom, release of calcium. Okay? All right, so this is just another picture that I just added for the vibes. All right, small quiz. Which component of the muscle cell helps to transmit an action potential from the surface of the myocyte to produce the end result of increased intracellular calcium? Bilal says C. Bilal is on fire today, mashallah. I don't want to say if that's the answer, though. He might be wrong. Anyone else? Saad also says C. We're going to go with C. The answer is C. Perfect. Okay. T tubules are what helps transmit an action potential from the surface of the myocyte to the end result of increased intracellular calcium. Okay, coming to the second part, actin and myosin myofilaments. Stick with me in this one. It becomes a little tricky towards the end, but we're going to get through it, inshallah. All right. So starting with what a myofilament is a protein fiber present within the sarcoplasm of muscle cells, and it's made of two things, myosin and actin. And these filaments are responsible for the contractile ability of cardiac muscle cells. They overlap, and when they contract, they move like this, which then causes the muscle cell or the whole entire muscle tissue to contract together. And when these actin myosin filaments relax, they move apart like this. And that is what causes the muscle tissue to relax and to loosen, if you will. Okay. So I want you guys to focus on this thing here, the A band. You see how it extends like this here? So the A band contains thick filaments. The thick filaments are these maroon lines that you can see over here like this. These are the thick myosin filaments, okay? And now look at the I band. The I band contains these green thingies over here. These green things are the thin filaments, okay? These thin filaments over here. So this whole thing is a thin filament. Well, these green lines, these are the thin filaments here, okay? So the thick and thin filaments, as you can see, they overlap, literally interdigitate like fingers, right? Like these purple ones or the maroon ones are the thick ones and the green ones are the thin ones. And they literally, intersect like this, right? The thin filaments are attached to the Z disc in a hexagonal manner. This is a Z disc. You can see they're kind of like arranged in a zigzag manner or a hexagonal manner. And the thin filaments are organized at the M line. This is the M line. The thin filaments are organized at the M line, also in a hexagonal manner. This diagram, however, does not represent that too well, but it looks kind of like this, to be honest. The M line resembles that to an extent. Okay, that's all you need to know for A bands and I bands for the rest of this lecture. All right, coming to the actin myofilaments, we're going to look into detail on what the actin myofilament is, and we're going to look at the myosin myofilament, and we're going to talk about everything else from there. Okay, keep in mind we're still talking about muscles here. The muscle components, the myofilaments that make up the muscle, okay? So we haven't gotten to how the contraction happens or how the impulse for the contraction comes and causes the contraction. We haven't reached there yet. Right now we're looking at the components of the muscle and what the components are that take part in the contraction process, okay? All right. So what is an actin myofilament? It's a filament, so it's a repeating structure of actin molecules and regulatory proteins. These proteins include tropomyosin and troponin. Troponin is made up of 3 subunits, troponin I, troponin C, and troponin T. And out of these 3, the most important one that you need to know is troponin C, which binds calcium ions. Keep this in mind, okay? So this is what it looks like. These actin molecules are joined together in a spiral type shape. And every now and then you have a molecule of troponin, which binds onto this thread that you can see. Let me go to the next slide. Yeah. So this thread that you can see that's really wound around this filament is known as tropomyosin. And this tropomyosin thread or fiber that you can see here, what it's doing is it's really covering these binding sites on each actin molecule that you can see. Each actin molecule has a little myosin binding site, which binds myosin. This fiber, tropomyosin, that's spiraling around is covering the binding site on each actin molecule. And this essentially is preventing myosin from coming and attaching to the actin. These troponin molecules are the regulatory molecules of the tropomyosin rope. Okay, so they sit on top of the tropomyosin and they're keeping it in place, essentially. Okay, so now let's move to myosin molecule. Oops, sorry. Okay, myosin molecule consists of 2 heavy and 2 light protein chains. You don't need to know what the light protein chains are, but you do need to know essentially one component, which is the head of the myosin molecule. These heads are called cross bridges, and they have an ATP domain, an ATP binding site, sorry, an actin binding site, ATP binding site. This is really important to keep in mind because we're going to look at how ATP is involved in this whole contraction process later. So this site at the head is where the ATP hydrolysis occurs. So now just keep this shape in mind. There's this long tail with 2 head-looking thingies, okay? Now this is how a filament looks. Each myosin filament contains 250 to 300 myosin molecules. Every resource that I checked was different, honestly. The lecture says 300, AMBOSS says around 250, other things said around 270. So let's just go with 250 to 300, okay? And this M line is in the center. Remember how we looked at that earlier? Now, just to indicate really what a molecule, an individual molecule of myosin looks like, this is one myosin molecule. So this is how it looks. This is the myosin molecule. This is the head with the ATP binding site, right? And that's what it looks like. A single myosin molecule with the heads. Okay. Oh yeah, head of the myosin molecule. Okay. And there's finally something called titin. Titin is also not super important, but it's important to understand the concept of it. It's not relevant later down in this lecture. But I'll just explain what it is. It's a type of coiled structure that acts as a spring. It essentially helps certain muscles to maintain contraction for a longer amount of time. And it's present and it connects the Z disc to the M line. So remember how we said Z disc was where the actin fibers were present, and the M line is where the myosin fibers are located. So the titin really just connects to, and it acts as a spring. That's all you really need to know from this slide over here. Okay. Hold on. Yeah. Okay, yeah, that's really just it. It acts as a spring. It's a coiled structure. Here's another diagram that I just added. It's everything that I've already gone over, but it just kind of summarizes it really well. And it's a little more neat than the other diagrams that I have. Okay, now coming to cross-bridge cycling. I think we're already halfway with this lecture. So cross-bridge cycling, what is it, and what are we talking about? Cross-bridge cycling is essentially how the contraction of the muscle really happens. So here you have the initiating stimulus, the action potential. It arrives to the sarcolemma. It's transmitted through the T tubules into the terminal cisternae and the sarcoplasmic reticulum, and it causes release of calcium ions stored in the sarcoplasmic reticulum. How this process really happens is known as excitation-contraction coupling, and we'll go into that in just a minute. But here, what we need to know is contraction. We're looking at the contraction process. We're not looking at the excitation-contraction coupling. We're just looking at the contraction itself. So here, our concern is not with how the calcium is released, how the stored calcium is released. We're looking at what happens when the stored calcium is released. Okay? So the calcium within the cell is increased because of the sarcoplasmic reticulum releasing calcium. This calcium goes and binds to the troponin subunit C. Okay, this causes a change in the tropomyosin fiber that we were looking at earlier, the rope that goes around. And this tropomyosin fiber changes shape, and conformational change is induced, and you know how earlier it was blocking the myosin binding sites? Now the fiber moves. Or let's say it just moves a little bit. So those binding sites are now exposed. So the myosin can now come and bind to those myosin binding sites on the actin. Okay. So here we have these blocked myosin binding sites. What happens? Calcium comes and binds to troponin C. These binding sites become exposed, as you can tell in this image. And naturally, myosin comes and immediately binds. Okay. So let's talk about the binding process and how ATP is involved in this. ATP provides the energy that is required for cross-bridge cycling. The ATP binds to the myosin first while the myosin is in its resting state. I'll show you a diagram for this in just a minute. So while the myosin is in its resting state and not bound to actin, ATP comes and binds to the myosin. This ATP is then hydrolyzed at the ATP hydrolysis site on the myosin head, and it forms ADP and phosphate, one molecule of phosphate. And this now causes the myosin head to take a 90-degree angle. Before, it was at a 45-degree angle. Now it's a 90-degree angle. Now this 90-degree angle myosin head binds to the actin molecules. Let me show you this. So here it's the resting phase. The myosin is resting. There's a 45-degree angle. ATP comes along, binds to the myosin head. It's hydrolyzed to form ATP and phosphate. And this angle changes from a 45 to 90. So now it's called the cocked head, I think is what it's called. Yeah. Okay. Now when calcium comes and binds to the troponin C, tropomyosin moves and it exposes these binding sites, right? And now it's exposed, the myosin binds immediately. What happens now is the phosphate is released and ADP is produced, and the ADP is released, and that is what causes the contraction or the shortening of the fiber. What essentially happens is the shape of the myosin head, which was initially 90, goes from 90 to 45. So it has this like pull when it, changes angle. And that change in angle is essentially what causes this contraction to occur. Finally, now the contraction has occurred, the ADP molecule leaves and an ATP molecule comes and sits. And that's how the whole process really happens. Now I'll go over it in words. 1 second. Yes, it's called a power stroke. Okay. So let me start from the beginning. I showed you a diagram. We'll go back to the diagram, but just to recap in words. So what happens? The myosin head binds to the exposed myosin binding sites on the actin molecules. This is known as cross-bridge formation. Phosphate is released. Once the myosin head is now bound, the angle of the head changes from 90 to 45, immediately pulling the myosin along the actin. This causes the muscle to shorten or to contract. This is known as the power stroke of the myosin head. Okay. After the power stroke, the ADP is released and a new ATP molecule comes along and binds to the myosin head. The myosin head then detaches from the actin filament and returns to its original position, loosening the cross-bridge. So this is known as the cross-bridge cycle. The cross-bridge forms when the myosin binds the actin, and then it moves. It causes a change in the shape of the angle. Then the new ATP comes and binds. The cross-bridge loosens. New calcium is released. This causes the myosin to bind again, 45-degree angle change, then it's loosened when a new ADP comes. The whole cycle keeps repeating itself. As long as calcium channels are present and energy is present, which is there in the form of ATP, there's always going to be a new cycle of cross-bridge formation. So coming back to the diagram, let me just go over it one more time. This is the beginning phase, resting phase. Myosin is not active right now. ATP is bound to it, and the angle here is a 45-degree angle, okay? Now, this is hydrolyzed to ADP and phosphate, and this 45-degree angle becomes a 90-degree angle, and at the same time, calcium comes and binds to the troponin C, which exposes the myosin binding sites. Myosin comes and binds immediately. This causes the release of the phosphate ion that was, produced over here. It's been here throughout. Now it's released. That's lost. Once that happens, the angle changes from 90 to 45. 1 second. Okay, so the angle changes from 90 to 45. And that is the power stroke part. Then finally, a new ATP molecule comes and joins. And that loosens the cross bridge. Any questions so far? We're done with maybe the most difficult part of this lecture. Congratulations, you guys. Let me know if you guys have any questions. I'm going to move on to the next part. Little pop quiz. What prevents actin and myosin from existing in a continuously bonded state? Someone said C. That is correct. The answer is C. Tropomyosin covers the myosin binding sites. Perfect. Now coming to excitation-contraction coupling. This is the part where I feel like, it really is just common sense, and it puts a nice, it gives you a nice perspective of what a cardiac muscle is like versus what skeletal muscle is like, because here is where the real difference comes in. Okay. So first, what is excitation-contraction coupling? It is the translation of an electrical signal from the sarcolemma into a muscle contraction. So we spoke about how the muscle contraction itself happens, but we didn't speak about where the impulse comes from or how the impulse ultimately causes this contraction, right? We mentioned something like impulse comes, channels open, calcium is released, and that calcium that is released from the sarcoplasmic reticulum goes to the troponin, binds to it, allows the myosin to then bind, and so on and so forth. Cross-bridge cycling, that happens. Excitation-contraction coupling is the process from which, or the process of which a signal first comes from the action potential. This signal is then converted into something which then allows the sarcoplasmic reticulum to release calcium ions. These calcium ions that are released then go to cross-bridge cycling, where they do the whole actin-myosin thing that we just discussed. So now we're looking at the process of how the calcium ions really come to be released. from the sarcoplasmic reticulum. Okay, so what happens? First, an action potential arrives on the sarcolemma. This causes muscle cell depolarization. This depolarization spreads across the surface of the cell membrane, across the sarcolemma, and into the T-tubules. This then causes opening of voltage-sensitive dihydropyridine receptors. Remember how we discussed that these were present on the T-tubules? So these voltage-sensitive dihydropyridine receptors on the T-tubules are coupled mechanically to ryanodine receptors in the sarcoplasmic reticulum. What I mean by coupled mechanically or mechanically coupled is that they're next to each other like this, kind of like a door. When one opens, the other is forced to open along with it. Or when one depolarizes, for example, in this case, the other is forced to undergo a conformational change because of the first. So they're mechanically coupled. They're next to each other like this, okay? Keep that in mind because that is an important detail later. So what happens? The mechanically coupled door, the DHPR, opens, and that allows the ryanodine receptor in the skeletal muscle to open. And this releases calcium from the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum stores calcium. This releases calcium into the sarcoplasm and increases intracellular calcium. This calcium now does what? Goes to the troponin C, binds to the troponin C, which then causes the tropomyosin to move, which then exposes the myosin binding sites. So the myosin comes and binds and causes contraction and so on. So this is how the intracellular calcium is released. Let's break down how it's released in this diagram. The depolarization comes and changes the membrane potential here of the T-tubule. Remember, this is still the T-tubule. The DHPR receptor is now positive. This causes a conformational change in the ryanodine 1 receptor, which now opens and allows calcium ions from the sarcoplasmic reticulum to now go out and go to the troponin C, okay? Now let's come to cardiac muscle. How is cardiac muscle different? It's only a tiny bit different, but that difference is a big deal. So in this case, it's almost identical to skeletal muscle contraction, except for one small thing. The action potential arrives on the sarcolemma. So far it's the same as the skeletal muscle. This opens the voltage-gated dihydropyridine receptors in the two tubules. Still the same as the skeletal muscle. Now, this causes an influx from the extracellular space into the sarcoplasm. So the difference here is earlier, those voltage-gated dihydropyridine receptors were only changing their potential, like they were only becoming positive, from negative to positive, but they were not allowing entry of calcium. Let me go back for just one sec. So you see how it's not actually opening. It's just being changed from negative to positive. In this case, the DHPR receptors actually open, which will now allow calcium ions to travel into the cell and travel to the ryanodine receptor, land on the ryanodine receptor, cause it to open, and cause it to release calcium ions into the sarcoplasm from the sarcoplasmic reticulum. Let me cover this in a image. Okay, so this is the image that I'm talking about here. This is the extracellular space. This is the T-tubule once again. This is the sarcoplasm, and this is the sarcoplasmic reticulum. In this case, the DHPR receptor opens, allows calcium to come in from the extracellular space, enter the sarcoplasm or the cytoplasm of the cell, go to the ryanodine 2 receptor. It's not ryanodine 1 anymore, it's 2. Bind to it, cause it to open, and now cause calcium to enter. So the difference here is what? What is the exact difference? Here, let's put them both side by side. In skeletal muscle, you have a mechanical coupling between the dihydropyridine receptor and the ryanodine 1 receptor. When this changes, when its potential changes, this causes a conformational change in the RYR1, which then causes entry of calcium. In this case, however, there is calcium coming from the extracellular space. The dihydropyridine receptor opens, then it causes calcium to land on the ryanodine receptor and then causes it to release more calcium from the sarcoplasmic reticulum. Someone in the comments or the replies, what is it called? Yeah, chat. Someone in the chat said that this is called calcium-induced calcium release, and that would be absolutely correct. Essentially, calcium is coming from the extracellular space. It lands over here on this ryanodine 2 receptor, causes calcium to come from the sarcoplasmic reticulum into the sarcoplasm. Okay. Right. Yeah. Now let's compare them side by side, I think, in more detail. I think a table always really helps when it comes to comparing things. So skeletal muscle, the DHPR is mechanically coupled. Cardiac muscle, it's not mechanically coupled. It needs calcium-induced calcium release. The ryanodine receptor needs calcium to come onto it and sit on it before it can release anything. In skeletal muscle, the calcium is not coming from outside the cell. It's only being released from the sarcoplasmic reticulum. Whereas in cardiac muscle, it was coming from outside. In skeletal muscle, they use RYR1, ryanodine 1 receptors. In cardiac muscle, they use ryanodine 2 receptors. In skeletal muscle, the T tubules are a little smaller. This is not really an important difference, but it was mentioned in your lecture, so I decided to include it. And in cardiac muscle, they use wider T tubules. In skeletal muscle, there's also more DHPR receptors. Again, this is not something important, but I just included it. And in cardiac muscle, there's fewer DHPR receptors. A few similarities between the two. Both are striated muscle. So they both have characteristic A and I bands with the same tropomyosin troponin action going on. The contraction coupling, the cross-bridge cycling process is the same in both. Both skeletal muscle and cardiac muscle contraction are initiated by an increase of Ca2+ or calcium in the sarcoplasm. Just the way that the sarcoplasm gets this calcium is different. In skeletal muscle, it's within the cell sarcoplasmic reticulum is what is releasing the calcium. Whereas in the cardiac muscle, this calcium is coming from an extracellular source first. Yeah, okay. Now a general comparison of skeletal muscle versus cardiac muscle, not to do with the excitation-contraction coupling specifically. Here what happens, so skeletal muscle fibers they all require to be stimulated individually at the motor end plate, meaning each neuron has to go land on the muscle cell membrane to cause it to contract. That's not the same in cardiac muscle. What happens in cardiac muscle is all the cardiomyocytes, as we mentioned at the end of the last lecture, they're all joined together very, very closely by these intercalated discs or these gap junctions. These gap junctions allow ions to pass through and the cells to communicate. So any impulse that comes is actually able to go through the cells one by one itself. They don't need to be stimulated individually. You don't need to do that in terms of cardiac muscle. They communicate between themselves to allow one impulse to arrive and to allow coordinated contraction throughout the heart. This is called syncytium or syncytium, I think. I don't know. I finished step 1. I still don't know how to pronounce this word. Syncytium, I think. Anyway, this is known as syncytium, which is the coordinated contraction of the cardiac muscle. This is possible because of the extensive gap junctions, which allow rapid transmission of electrical impulse. Okay, this is a slide from your doctor's lecture. Essentially, the most important thing that you need to know here is that in the skeletal muscles, there are more dihydropyridine receptors and ryanodine receptors, and they're arranged closely together because remember how they're mechanically coupled? So it's very important for them to be in a specific order. Each dihydropyridine needs to have a ryanodine. Each dihydropyridine has to have a ryanodine. Like there has to be some systematic order for these. And so there's more dihydropyridine receptors here. However, in cardiac muscle, the dihydropyridines do not have to be linked mechanically to each ryanodine receptor, so there are fewer, and they're arranged in a more scattered way, almost random. Okay, I think we're done with this lecture. Actually, we just have a few questions which I wanted to add. This question is a little tough; it's a little long, so I understand if you may not want to do it. But I'll give you like a minute or 30 seconds to see if you can get it, and then I'll tell you. Okay. So someone has said B. Any other answers, guys? Come on, quick. I want to wrap it up. I want one more answer, one more person to attempt. It's okay if it's wrong. Okay, let me proceed. The answer is B, correct. Let's go through it. So B starts with number 3. Number 3 is the spread of the impulse over the sarcolemma and the T tubules. That's the first thing that happens. The next is number 4, which is the change in conformation of the DHPR receptor, directly causing opening of the ryanodine receptor by mechanical coupling. Because remember, we're talking about skeletal muscle here. And the final one is number 2, which is release of calcium from the sarcoplasmic reticulum. And in case of a V, or 5, if you will, that's that never happens. Actin never binds to troponin C. And what else are we not using? We're not using number 1. Number 1 is what happens in cardiac muscle, not skeletal muscle. So, okay. Another question. Which of the following causes the power stroke phrase in cross-bridge cycling? Come on, this question should be easy. It's pretty simple. What do you guys think is the answer? Which of the following causes a power stroke? Is it A, detachment of the myosin head from the actin filament, B, binding of the calcium in the troponin, C, changing of the angle of the myosin head from 90 degrees to 45 degrees? I don't know why it looks messed up. Let me fix that. Or is it D, the myosin head binding to the exposed myosin binding site at the angle? The answer is C. Okay, perfect. And that's it. Any questions, guys? And before you start leaving, this is a QR code, which is for feedback from me. I would love to hear from you guys for my own improvement and, to help me become a better tutor, inshallah. So I would love for each and every one of you to fill out this form. Don't worry, it'll be anonymous. I promise it is. But I really want to know just, what you thought of the lecture and what I could do to improve, or generally any comments that you might have. And yeah, do you guys have any questions? Someone in the chat asked, do I have any advice for question banks and stuff like that? Any particular resources that I would recommend? Okay, so I would definitely recommend that you do questions. I think it's really good that you're already interested in, that you're already thinking about doing questions. That's a very good sign. I would definitely recommend looking at past exams. That's maybe the number 1 most important thing. So each batch collects past exam questions, right? Do not underestimate the importance of those questions. They're very, very helpful. You can go all the way back to 11th batch, for example. I'm 11th batch. You can go to 12th batch. You can go to even 10th batch, for example. All these people collected their exam questions, and those are really helpful when it comes to studying, because they really make you understand what your professors are looking for. That's the first thing. Second, I would really recommend the AMBOSS question bank. I know that AMBOSS is a paid resource. I definitely believe that paying for it is very much worth it. I don't believe there's any resource that's quite like AMBOSS. AMBOSS questions are probably the make or break of everything. Like when I was giving my step 1 exam, I used AMBOSS a lot. It was very helpful in some cases. Although UWorld, the actual question bank that you use for step 1, I also used that. But some things when I didn't understand, AMBOSS was always my go-to. It was very helpful. What else? I think for physiology, what's the physiology book that you guys use? Guyton. Yeah. Guyton Physiology has a bunch of questions as well. I think there's it's called the Practice Questions Handbook or something like that. I did practice questions from there for CVP when I was in first year as well. And I think that's all the resources that I would recommend. if you guys have any other questions, please do let me know. And yeah, any other questions? It doesn't have to be related to the lecture. Take your chance.